My name is Mike Dolinsky. I'm with Taurus Ag, and we're going to look at the second part of our series, Plants Know What to Do. And we're going to talk about wheat in terms of what wheat knows to do. If you haven't looked at the first episode of this, part one on biology and functions in the plant or processes in the plant, you probably should do that before you move forward on this. For those of you who have had that, I hope you've recovered from it. It was pretty intense, and this one is a whole lot easier, in my opinion. I'm going to just make a few comments about wheat, not a whole lot, and then we're going to go right into it. Germination and emergence, rapid growth, reproduction and seed development, grain fill and senescence. I guess it's safe to say that wheat was my first love after retirement, after my wife. And once I got into looking at wheat, uh, because of an insect in wheat called the wheat midge, and I was photographing some of this stuff in the field. I got my microscope and I started looking at wheat in a really intense way, and I've accumulated what I think is a phenomenal portfolio of photographs of wheat and how it grows. And I want to share that with who's ever on this webinar, and whether it be a student, a farmer, a retailer, and so on, a seed grower maybe. Just to sort of get from my perspective anyway, and I'm no breeder or anything like that. I just take pictures and then I read a whole pile of papers about how to understand what I've seen. So let's get on with it. Now, wheat was the foundation of civilization, and wheat was the first crop that humans started growing and storing so communities could be set up. That sort of started in the Middle East, as far as we can tell, about 11,000 years ago. And wheat has changed since the beginning through selection, just as corns had changed from its wild type. So it's a really interesting crop. You saw that the roots are great, the genetics are great, lots of diversity in terms of genome. And today we grow wheat almost everywhere in the world, all kinds of different wheats. I'm primarily talking about spring wheats. I'm not going to really talk about winter wheat or soft wheat or durum wheat. Uh, in my view, probably some of the nutrients vary, and I'm not a real agronomist in terms of running into those kind of things because I live here in Edmonton, Alberta, where we only grow spring wheat, so I don't have a lot of access to growing those other crops, and I don't deal with those independent of how I talk about wheat. Talked about how the plant responds to all kinds of abiotic and biotic stresses through a series of signaling and genetic modifications to get the plant into homeostasis and deal with stresses and become tolerant to them as best it can, just like all eukaryotes do. And I all know what eukaryotes are now from the last episode. So the same thing is going to happen today. I'm going to just expect that we understand that when plants are making changes, they're doing it genetically because they're sessile, they can't move. So they have to do everything by up and down regulating and expressing and non-expressing genes is the other way, but I like to simply say up and down regulating genes, and I hope folks understand what I mean by that. And they produce products, biosynthesize these products, and then move them around to do things. Proteins, enzymes, hormones, those kinds of things. And the last thing I'll talk about in terms of review, because when we get into grain filling, it's really important that you understand where the nutrients that go into the grain are coming from. And it's basically uh, the cells. The cells, as they're aging and so on, will be lysed, broken down, and the nutrients extracted. And it's important to look at this because look at all the nutrients in there. Look at the chloroplast, copper, calcium, magnesium, iron for electron transport, and then the mitochondria with the same thing, iron, copper, and so on for electron transport, and the proteins in there. The vacuole is the storage cell where you break down the nucleus in the end, the peroxisomes, and the endoplasmic reticulum, all these organelles, and that's all you have to remember, these organelles that are in there are broken down. And then the uh, uh, the system here, the river, the endoplasmic reticulum can move those nutrients when they're being broken down between cells and through the plasmodesmata out of the cells as they're being broken down and move them around in the plant. And we just have to believe that the plant can do this, and it does it genetically. I didn't mention much about nutrients in the last presentation, talking about systems in the plant. And I'm going to be talking about wheat, canola, and peas in this series. So wheat is the first one we're talking about. But we'll take a look at all of these three major crops here in Western Canada, uh, because that's where we basically do our business, from Ontario to British Columbia. Because it's kind of easy to compare them when we start out early in the system like this. And we'll look at the macronutrients and then the micronutrients. But if we take a look at one crop specifically, you'll see that canola uh, is a big user of nutrients. It takes up a lot of nutrients and exports a lot of nutrients to the urban feedlot and the livestock feedlot. So let's take a look at nitrogen. Look at this, 3.3 pounds of nitrogen per bushel, a lot more than anything else. Peas do as well, but they get a lot of that from nitrogen fixation, and the peas send off a lot of that in the removal, in the seed. Now, and that's the objective of the crop we grow, is to produce seed. And it's the nutrients in the seed that we want to have. So we got to get them out of the soil and into the plant. Phosphates. Look at the uptake of phosphate in canola. More importantly, look at what goes into a pound of phosphorus per bushel leaves. That's, oh, a lot more than anything else. Potash, potassium, not much difference, although peas take up a lot more. Sulfur, canola has got a lot of sulfur in it, uh, and it's got a lot of nitrogen in it because it produces high protein. And I forgot to mention that if you can use an easy rule of thumb: the higher the protein content of the seed or the plant, the higher the nitrogen requirement. And because the amino acids that make up, or the amino acids that make up the proteins are a combination of those that have nitrogen and sulfur, because of the amino acids, two of the amino acids of the 2022 amino acids that are used in eukaryotes, uh, two of them contain sulfur, cysteine and methionine. So there's always a ratio of nitrogen to sulfur in order to get good protein. Just putting on a lot of nitrogen when you have a sulfur deficiency may not help you. It may hurt you, because the plant can't make the amino acids and the proteins without the sulfur. Calcium is highly regulated by the plant and used as a signaling agent and used in cell wall structures. But you can see that, again, canola takes up a lot of calcium, and it also has a fair amount in the seed. Magnesium, our good friend canola. I always like to call canola a plant that grows like a weed. It can extract nutrients better than anything else, even relatively compared to wheat. It's got exudates that are much more potent. But we have all of these nutrients here that are being picked up, the four macros, the semi-macro nutrients here, or secondary macronutrients. So we have a various distribution of nutrient uptake. So the message here is that, and farmers know this, you don't fertilize every crop the same, because the needs of the crops vary. With peas, for example, they fix nitrogen, so you don't put on much nitrogen, if any, when you're growing them. With wheat, you just put down, if you're not soil testing, then you can just put down what you think is going to be removal. But generally, you would plan your fertility program around a soil test telling you what's in the soil, taking a look at what you think your yield potential is going to be, uh, putting down enough nutrients to cover what's going to be taken up, and that sort of keeps you at least in position zero, so you're not mining your soil. If you have an opportunity to add more, the nutrients that are immobile, like a phosphorus or potassium, when prices are low, you can add more, and the residual will stay there in the soil for plants to use in the future. Now we'll take a look at some of the micros, and we can see that zinc is used, when you look at all the other micros except for iron, used at, and a little boron here, but boron's kind of an odd Boron can act as a metal. It's called a metalloid, so it can go in all kind of directions and switch depending on factors in the soil. But on the whole, canola extracts a lot more than anything else we grow. But you can see here on the zinc, they all use a lot of zinc. That's why it's an issue worldwide with about 30% of the world population, human population, showing deficiencies in zinc. Manganese, wheat uses a little more manganese, a lot more than peas do. And manganese is key in terms of photosynthesis and breaking that water molecule, and also stress management. So we always want to get our manganese there, and peas seem to get along nicely with a little lot less. And that's the beauty of it. These plants need different amounts to function properly. So just because one crop uses this much, it's not safe to assume that another crop will use the same amount. Copper. Now here's an interesting thing here between canola and copper, and we grow about 22 million acres each of wheat and canola here on the prairies. And a lot of people expect that if they're going to have a copper deficiency, they're going to see it in wheat. And yet, when you take a look at this, canola uses as much copper on a per bushel basis as wheat does. And yet, we never even talk about copper deficiency in canola, but we do talk about copper deficiency in sandy soils across Western Canada. But it is interesting that all plants need copper for that electron transport chain we talked about in photosynthesis and mitochondria. Boron, you can see one thing that's important. It is generally a rule of thumb that dicotyledonous plants use more boron than monocots. In this particular case, canola uses a whole lot more and uptakes a lot more and takes away a lot more than wheat and peas. And lastly, iron. Again, you can see that canola pulls a lot of iron out of the soil and moves a lot into the meal because it's an oilseed crop. So cattle probably get a good supply of iron from the meals that they're consuming. Whereas wheat, it takes up iron. All plants need iron for all those processes in mitochondria and photosynthesis. So, what I talked about before in terms of all these nutrients and their role in plants, you can see that these micronutrients and the transitional metals, except for boron here, are really, really important, and the plants recognize it because it's my belief that the seed tells you what the plant needs, and if you know what the level should be, it tells you what the plants aren't getting. So it's a way to measure whether you're actually feeding your plants properly. And I'll talk about some of the metals and nutrients that we use during the growing season to support plant health. So we're getting down to the nitty-gritty here. This is the typical growth curve. Plants all basically grow the same. They grow in this part here. It's called the vegetative stage, rapid expansion. So people think, well, it's growing leaves, it's growing roots. Well, that's true. But at the same time, it is during this period that it's growing all the reproductive parts, which is the key thing. The plant only needs the leaves to support growth and fill the seed. The plant is only interested in growing seed to carry on its progeny and its genetics into the next generation. So it's this part that grows the plant, and it grows it and fills it and is the sink for production from photosynthesis until it reaches reproductive stage. When it hits flowering, suddenly the sink becomes the seed. So the plant then focuses its photosynthesis in terms of filling and finalizing the seed and cuts off some of the supply to the roots. It has to, unless it's got phenomenal conditions, it's got a partition where its nutrients and photosynthesis are going to be concentrated. After that, it starts drying down and filling the grain and moving everything from here into the grain. And you can see here, this just depicts what happens. The plant is growing, picking up nutrients. You can see phosphorus is sort of steady. It uses a lot of the phosphorus early on. After here, it's sort of a maintenance level because it's only interested, the plant is only interested in the organic phosphorus. So it doesn't disappear. It's in the cells and can be sort of recycled, for lack of a better term. You've seen that in terms of ADP and ATP, that cycle that goes on in the plant. It reuses some of that inorganic phosphorus. And then it pretty well levels out here in terms of uptake. And then the filling is related to remobilization. Now, I want to just show you a little bit about a spikelet in the seed head, because we're going to get down to the nitty gritty in terms of what actually goes on inside the spikelet. So the spikelet here, which usually in wheat will have 4 mature seeds. If you get 3 mature seeds, that's an average crop. You get 2 mature seeds, it's a failure. You get 4 seeds, it's a bumper crop. I've only seen 5 seeds in a spikelet a few times in the field. And each one of that group of seeds we call the spikelet sits on a rachis, which goes down the center of the seed head and feeds each one of those florets. Now, each one of the seeds here you can see here's the embryo, here's the stigma, is covered by or surrounded by something called a lamella on the one side, on the bottom side, and a pellia on the top side. In other words, it's protected. And then there's 2 glumes that surround the whole base of that spikelet, and that sits on the rachis. Now I'll show you what that really looks like in real. So here's a wheat head, and here is that group of florets called a spikelet, and you can see this one has 4. 1, 2, 3, 4. These 2 will be the same size at the end. This one will be a little bit smaller, and this will be the smallest. And that spikelet is sitting on this rachis node. So the plant is bringing up nutrients here, and it's partitioning it off to each one of these nodes as it goes up the plant to feed each one of that set of florets. So what does this mean? Well, take a look at this primary seed head and look what you see. 4s, 4, 4, 3, 4, 3, 2, 2, 4, 3, 3, 3, 2, 2, 2. What happens is that as the seeds pollinate, and the pollination wheat starts in the middle of the head, the majority of the nutrients in the plant are available to those initial seeds that are pollinated, and therefore you get the 4s usually in the middle, and you'll get the biggest seeds as these base 2. And in the tiller, notice what you get. This was a really dry year back in 2020. You get next to nothing. And these seeds are all smaller. So you want to get after the growing more of these seeds. And just think about it. If you can grow 1 more seed in each one of those spikelets, and the millions of plants you have out there, and the million of spikelets you have, wow, that's where you get your bumper yields, when you get a whole bunch of 4s, or maybe the odd 5. So we're going to look at this growth curve from here, from germination through rapid expansion to grain fill, and cover off why you get a crop or why you don't get a crop. Let's look at germination. Seed is really important. I don't think I even realized just how important seed is to success of your crop. And we'll go through seed here, just so you can see how a seed actually grows or germinates. And sometimes I get things mixed up. I call an embryo germ, so you'll have to forgive me for that terminology. It is something that I habitually do. So anyhow, here's a seed that I cut in half. It was just germinating, so it worked out quite well. So here's the embryo, and you can see here is what is called the coleoptile. And right here is the first true leaf. Okay, and you can see that the seed is still solid. This is the starch called the endosperm, and it's all mixed with proteins. The proteins mix in with the endosperm. The endosperm is starch, and all starch is a whole bunch of sugars put together. And there's 2 kinds of starches here, amyloplast and amylopectins. And they're different sizes
so that the plant, when it's going to use all this nutrient here, has small kinds of storage cells, medium-sized storage cells, uh, storage cells, and big storage cells. So it can extract the nutrients over a period of time, because this nutrient packaged in the seed, in a wheat seed, will support the growth of the uh, plant or the shoot for the first 10, 12 days of its life until it can photosynthesize, because until it can photosynthesize, the plant can pick up nutrients with the root hairs, but it can't produce any sugar. So it has to depend on the starch that is stored in the wheat seed. Now, this is all sort of dead tissue here. It's just sugars. Now, what's alive is the embryo and this little line here called the aleuron that runs around. You can see this fine little red line. That's all that's alive. And this is about 3% of the whole seed. So that's kind of important. But this is where the mitochondria survive from one generation to the next, and the plastids survive right here. And I'll talk to you about the importance of that. Now, as the plant imbibes water, it has to increase its size by about 40 to 50% to stimulate the new growth and the germination of the seed and the process of uh, getting these nutrients available and getting the system going. And I'll go over that in a while. But let's take a look here. This is called the scutellum. It's at the base of the embryo. So that the scutellum is the sort of the tissue that regulates the flow of the sugars from the endosperm into the growing new shoot and root. So that's basically where we're going to go from here. Now, if we take a look at when the seed is germinating, uh, we see a couple things. This is called the coleoptile, which is going to support the new shoot coming out of the ground. And you can see this one's a little smaller, and as it grows, it gets bigger. This is the radicle, which becomes the first root, and you can see here it is growing, and it's already got root hairs. These are seminal roots, and these haven't got any, and this is called the coleoresia. So you don't have to worry about that one. But that's generally what happens here, and wheat has 5 seminal roots, so to speak. So there are a lot of things that can affect seeds. When they're growing. Fertilizer salts, right here. You can get toxicity from fertilizer salts. You want to have these kinds of things growing here where we're using, the roots cannot be affected. We can have damage from drying, salinity, cold soils, slow up germination and allow microbes to take effect. Poor seed. If you got poor mitochondrial survival, you got poor seed. It's going to be weak seed and it gets damaged. Damaged seed, during combining and transport, seed germination can be reduced by damage to the germ at the end of the seed. So there's a lot of things that can be going on. So you should sort of look after it. And I'm a firm believer with the company that I work with. We use a seed treatment, a nutrient seed treatment, and we get really great success year in and year out. And for a while, I couldn't figure out why we can get such great responses. And now I kind of understand, by putting that on the seed, we can actually supplement the missing nutrients in the seed and allow that seed to really take off. So let's take a look at what's in the seed. Well, we got sulfur, and this is what's in the embryo. This is what's in the endosperm, and this is in the seed coat. A lot of the stuff in the seed coat doesn't get actually used. It's just left behind. And farmers that are been out in the field a lot, and when you go and dig up plants and look at the seedling, you can find the seed there, and there's nothing left in the seed but the coating, the coat. So the plant extracts everything out of there. But it's important to look at what it puts into the embryo. Look at the zinc. Remember how much zinc it uses? Well, zinc is involved in all the enzyme reaction groups in a plant and involved in stress management. And look at the amount in the endosperm. So it feeds the plant so it can deal with its genetics that's used to open up the pathways for messenger RNA to actually make copies of the genes. copper in photosynthesis and in mitochondrial activity, manganese in the same thing. You can see that the plant is geared up with all these micros and macros to start it with the embryo and feed it with the endosperm. And these give you clues as to what are important. And when I talk about zinc deficiency and iron deficiency, those are things that I personally focus on a lot, mostly about zinc, because we have quite a bit of iron just normally in our soils here. So these are the key nutrients here that we want in the seed: nitrogen, the proteins; phosphorus for energy and strong roots; potassium drives everything, stress tolerant; and all these micronutrients. Let's go a little further, because seed is pretty intriguing. Here's some work that Dr. Chakmak out of Turkey has done, and looking at staining so we can see where the proteins are. You can see the proteins are scattered all through the endosperm here. We got the zinc is in the aleuron here, and the germ is really highly concentrated. The iron's the same thing in the germ and here on this aleuron, depending on the angle you're looking at. This is another one showing the zinc. And the variance in the concentration depending on where you are in this embryo going here with the coleoptile in the root. But to me, this is the most intriguing photograph, and this one isn't mine. I'm not sure where I got it actually. But if you look at manganese, which is showing you where it is, it's in the embryo and the aleuron, not all the even the way around. It's right here close to the embryo, because that's where the moisture is coming in. That embryo is right there. If you look at the germ, and I'll show you what that looks like. Here's the iron, same thing, the embryo, and along the aileron; copper, same thing right there; and zinc. Look at the load in the embryo. It's just what we just saw looking at the nutrients in the seed. Tells you the importance of that stuff in the plant. And if we're getting deficiencies of zinc that are showing up in a wide scale, we're going to have deficiencies in seed, and we're going to have deficiencies in the embryos, and we have potentially a reduction in the vigor of our seed. Interesting theory. And I'll show you, or I'll tell you about a study I did with 2020 Seed Labs back in the year 2020, it just so happened. And I was dealing with Sarah Foster over there, and we thought we'd take a look at whether we could see any nutrient relationship to vigor and germination. So what she did is she selected 100 samples from the work that she had done in terms of customer germination vigor tests, and took a mix of seeds that were, high vigor, low vigor, high germination, low germination, and we crushed them all up, sent them to A&L Seed Labs, and we extracted the nutrients, and lo and behold, what did we find? 100% of them were deficient in boron, 54% of them were deficient in zinc. The boron I kind of expected. The zinc I sure did, not from Alberta. I expected that in southern Saskatchewan and the brown soil zones, but not here in Alberta, where I think we haven't got such grass originated kind of soils. Some in southern Alberta for sure, but not more in central and sort of northern Alberta. So that sort of supports the theory that deficiencies of zinc are becoming a problem, and that's going to reduce the vigor in my estimation for sure. And I'm a big fan of big seed. I think, we get more seminal roots, we get faster growth, and we get more fertile tillers. But most of the data that comes out of research says there's no difference whether you're a small seed or a big seed. It's a matter of what's in it. And there probably is some truth to that, but I think there's a much more nutrient in there. Here's seed developing. You can see in this spikelet, these two will always be the same size pretty well, and this one's bigger and so on. And this one gets the nutrients first because it's sitting on that rachis node. And we can see that in the actual production of the seed, and we see it here. And I think big seeds are good. I frequently tell farmers, if you're cleaning your own seed and you're not buying pedigree seed, which I'm not suggesting you do that, but just clean it really hard and go for the big seed. It doesn't cost you a whole lot more. So how does seed really get germinating? Well, the first thing it does, it takes in water. Right here, it's through the germ because that germ is kind of porous. It gets rid of abscisic acid. It breaks it down because that's what put it into dormancy, and it generates gibberellins, which stimulate growth. That stimulates the aleuron to produce uh, alpha amylase, which breaks down starch so that the plant can get glucose or sugar. It also produces proteases, which break down proteins. So it starts breaking this down and feeding it through the scutellum to the embryo that's germinating. And here is alpha amylase. Uh, this is chloride, and this is calcium, and these are proteins. So we need a chloride in here in this process, and we need calcium in alpha amylase to run this process. So those are some of the key nutrients in there. I showed this in the first presentation I made on processes because this is really, to me, quite fascinating. When the plant or when the seed starts taking in water and we get some moisture in the seed, because you can't move anything without water. So we need that. So it starts leaking out solutes. It starts taking the solutes that the mother plant had put out there and makes precursors to repair mitochondria. I've already mentioned, but the mitochondria have to be transported with the seed. Otherwise, the seed won't germinate and won't grow because it can't biosynthesize mitochondria. Mitochondria have to be multiplied. They multiply by binary fission, just growing and splitting in half and so on. So it has to take the remaining mitochondria that were shrunk down, repair them, and then it starts multiplying. So we get synthesis starting up here. Then the proteins are synthesized using existing messenger RNA that was also provided by the mother plant in there. So we can start protein synthesis and we can produce new messenger RNA to support growth. So there's this rapid pickup of activity, then a flattening out of that activity in phase two as the plant is fixing up everything, fixing its DNA, fixing its mitochondria, getting ready to really grow. And the plastids are also having to survive in there, the chloroplast or the protoplast, which makes the chloroplast in the end. And it starts repairing the DNA. Then we get cell division and off we go. Stored reserves are mobilized and the radical cells elongate. And when the radical breaks through the coating on the germ end of the seed, the embryo end of the seed, germination is over. So take a look at what happens when we put nutrients on a seed. These are seed that I got from Chinook treaters. You can see, look at how the concentration occurs in wheat. You know, if you look closely at the germ, it's kind of flaky. It's really thin. So I think it seems to hook on, and I see this all the time. It hooks on to the germ end of the seed. And when the plant is starting to germinate, it gets nutrients by extracting it from the root hairs that are here, but it starts getting nutrients as soon as it starts taking in water as the nutrients on the seed solubilize. So each seed then has an opportunity to supplement the existing seed nutrients in every seed. That's how we equalize the ability of seeds to survive. And I think that explains why we get such really good responses to active prime. It's a small dosage, but relatively speaking, on the seed, it's a large dosage, and the seed can get it really quickly. And you take a nutrient like zinc, when the seed is in the ground, it's cold. The roots can't pick up anything quickly. So the only way that we can get some more zinc into the seed is through the water that is carrying the zinc into the seed as it imbibing water. It may seem like a small amount, but that's why they're called micronutrients. So here we're going on. The seed is going to germinate, and here you can see it's expanding. This is the coleoptile going up in that first true leaf. Look at that. It's already there. The other thing that I will point out is the actual growing point is right down here, right there, that little tiny point. And that is going to be what produces the seed head. As these kind of go up, it'll produce some more leaves, and then it'll produce the seed head real early. Now, here's the coleoptile coming up with the first true leaf inside of it. And remember from that first presentation, I said that the chloroplasts are not chloroplasts until they get sunlight. Okay, so see what I mean? They come up yellowy and clear there, and then they're off and running, and I'll show you what comes up next. At the same time, the seed is putting out 5 seminal roots to support it, and then eventually it goes on and produces additional roots from the crown node. But back here, we can see that now the first true leaf is green. So it is now photosynthesizing, and if you look at it, you can see the stomata, these little white spots here, or excuse me, are the stomata, and they are now photosynthesizing, so the seed is now free of supporting the plant. It may be pulling some, but once the first true leaf is out and going, it's independent. And you can see that the first true leaf is always recognizable because it's a blunt tip on it. We're getting into rapid growth. Here's leaf number 2, leaf number 3, leaf number 3, and by now, going into the 4th leaf stage, the seed head is soon going to come above ground. The seed head is down here, and the growing point is down below ground, and the seed head is formed. It's probably right about here. And we'll talk about tillering for a minute, and then we'll get back to how the plant goes. So tillering is just a way that the plant can grow more plants. And you can see here we have a wheat plant. Here's the intercrown node. Here's what's called a coleoptile tiller, and you can see these quite frequently. And I'm not sure if there's a good reason why a plant will do that. I don't know if it's stress or I haven't really looked it up, but this will put out another tiller. But most of the tillers come from the crown node right here. And then as soon as you get the crown node going, of course, it's like the plant growing another whole plant. So it starts putting out nodes here, crown nodes, like corn puts out brace nodes to extract more nutrients. So here's the coleoptile tiller. Keeps coming, and then as the tillers, here's 2 tillers on it. That's about all you want on any kind of a plant, because if you put out any more than that here in the prairies in this dry, you're just wasting the plant's energy on that 3rd tiller, or 3rd tiller, and the plant will probably kill it and extract what it can out of it and get on with life. And you can tell the tillers from this profile, which is like a coleoptile on the tiller. Keeps on going. Excuse me. Here's the seed head emerging from the ground at 1 centimeter above the ground. First node is right there. Here's the second node at 2 centimeters. It's now getting bigger. And here's the 3rd node at stage 3.5. The seed head is always above. This will be the shortest part of the stem. This will be the second shortest. And this extension from the 3rd node up, called the peduncle, will be the longest. When we move in the reproductive stage from here on in, we'll get a big shift in terms of demand for nutrients. It's a little early to what you've seen so far, but as we're getting closer to pollination and so on, we'll see a big move on all these nutrients expanding and moving into the reproductive parts of the plant. Again, the sink now becomes the seed, so the plant is preparing to support that seed. This is just a shot from a book, or no, it's a website in Britain, which shows very nice photographs, but I'll show you what mine look like. And these are just showing the spikelets growing on the seed head at a very early stage. So here we go. This is at stage 33, which is the one I showed you, the 3-leaf stage, 3rd node stage, sorry, just emerging from the ground. Here are some more modern ones with my better microscope. You can see these are all the spikelets right here. Showing at a closer, these are the spikelets. So those are all going to keep on growing to become 4 or 5 actual seeds. And here's the tip. which is forming all the new cells. And these are the palea and the lemma that are growing to protect that seed and encase that seed. Here are just some shots of the same thing. You can see here's the awn, which is on the, I can't remember, I think it's the palea side, and there's the actual spikelet growing inside there. I throw this in because it kind of fits here. And this is a node on a wheat plant, and you can see that there's the node, and then there's the vascular system running up through the node. Well, I always wondered how a plant stands up after it's been knocked down by a windstorm and a hailstorm. So I decided to take a look, and all you farmers have seen this. You'll go down, and it'll be the last node that is going to take and pump that seed head up. Well, if you look inside, what you see is that the actual node has expanded on one side and pushed the actual seed head up and not grown on this side. And the plant knows that, because to stand upright, it has to have a mechanism, a geo-recognition mechanism in it, and it uses granules of sugar to actually sit in the node so it knows which way is down and which way is up, and it modifies it by expanding the bottom side, pushing it up. I thought it was just really cool. I never wanted to know how it did it, but it's really cool. Reproduction and seed development. Now, this is the beginning of heading stage 51. Well, I used to do a lot of staging and teaching, but that takes too long to get through this presentation by doing them all. And then ending by heading and inflorescences fully emerged, or the head coming out. So I should point out that the period from 20 to 30 days before flowering to 10 days after is critical, and there's a lot of mortality that takes place either running up to pollination or shortly after pollination if the plant is short of photosynthetates to support growth and plant health. And like I said, if you get 3 seeds in a spikelet, that's sort of an average crop. 4 seeds is a big crop. 2 seeds is a crop failure kind of thing. It's just that simple. The difference is 1 seed per spikelet. And this is kind of the way it goes. We saw this little tiny seed here, or spikelet. We'll go into the white anther, green anther, yellow anther, booting stage, heading, and anthesis. And as it progresses through all this, it's pushing that seed head and elongating that stem to move that seed head out of the boot. Well, here we are showing the stigma and the seed before the head comes out of the boot while it's booting. And then you can see that here we're getting, the boot is starting to open up. Here we're at stage 51. You can just see the tip of the head coming out of the boot. This legule protects the head as it's coming out, so dust and stuff doesn't fall into it. 53, it's a third of it out. 55, half out. 58, poof, it's out. So now what happens? At farm meetings, I always say, well, now what happens? And most everybody says, well, it pollinates. Well, not so. Now it pushes this head way up. It keeps pushing and expanding this head and maturing this seed head. You can see here that this one is way out here, this one is half out, and they're at various levels. Eventually they'll be all out, waving in the wind. And this is the way it works. You can see in sequence, this comes out, pushing, pushing, and pushing. And this push, it'll push out, if the weather's just right, it'll push it out at about 2 inches a day. And then it'll pollinate. So by the time you see the seed head out, and you're looking at fusarium or something like that, you should be getting your sprayer ready in about 5 days, being ready to make a decision if you've got the environmental conditions and the right staging to bother with a fusarium spray, for example, or maybe a wheat midge spray, although wheat midge isn't much of a problem with genetic changes we've made in wheat. So that's kind of what's going on. And then we're going to get pollination. So I'll give you some diagrams. Well, this is a photograph I made. Here are the anthers where the pollen is made. Here's the stigma, which is attached to the seed. And then the pollen will have to grow a pollen tube and grow it down and fertilize the seed. So that's the way it is, and you can tell what's happening by the colors. This is uh, uh, early on. You can see this is the seed here, this is the stigma, and the anthers are green. And if you take it out, you can see that the anthers are attached to the base of the seed. And they continue to grow, and then you can see that the anther is starting to slightly yellow, and it's starting to split here in the middle, and the stigma is starting to load up with a lot of sugars, hormones, and so on to support and prepare for the pollen, the male component, to arrive and support its growth, because the pollen tube has to eat its way down into the seed to deliver its nuclear material. You can see as it matures, the stigma spreads out so it can easily capture the pollen, and the anthers start to yellow, which is telling you it's maturing. You can see here that now it's yellowing and the tip of the anther opens, and you can see the pollen is starting to float out. You can see this is shining because it's got all that sugar on it and moisture, because it's got to eat its way down the transmission tissue. Well, when the plant is going to give up the pollen, it dries it, so it will be attracted because it's dry, and the stigma is moist, so it'll be attracted to the moisture in there, and it'll latch on to the stigma. You can see all the pollen here on the stigma. If you get in real close, you can see here that the actual anthers have given up the pollen, but the stigma has all these little spiny things, and the same thing is true in corn. I've got close-ups of corn that look like that, and the pollen sticks onto it, and then it grows the pollen tube down here, and then it'll grow it all the way down to the seed and release its nuclear material. I just threw this series. This just shows the pollen coming out. It's sort of emptying there, an empty one there, and then it exudes. It just tosses out the spent anther, and it looks like this. So these anthers are now empty. The plant has kicked them out, and it does that very quickly, usually the morning after pollination has taken place. And here's the this is sort of the first stage of pollination. You can tell now that the plant has been pollinated on these particular florets. And then we're completed pollination. And when you look at the anthers, these are the ones that have been recently kicked out because they're still yellow. These are a day earlier, and the plant is pollinated in about 4 to 5 days, depending on the weather. Diagrammatically, this is canola, but it demonstrates the same principle. The pollen lands on the stigma, grows a pollen tube down here. Here's pollen. You can see that it's enclosed, and there's an opening here called a germ pore, and that's where the pollen tube comes out. The pollen tube then grows down through that transmission tissue, and it eats its way through, basically gets in here through the micropyle, gets in there, bursts the tip, and the nuclear material pollinates as a double pollination here of the egg cell and the polar nuclei, which forms the endosperm there and the germ here. So that's where the two pollinations take place. And here's the pollen tube. It's the other single cell. They said root hairs and pollen tubes were single cells. And you can see it's got its own vacuole with transporters in here. So as it's feeding through here and moving through that transmission tissue, you can see there's a high concentration of calcium here and a lot of transporters that are taking nutrients from that transmission tissue that the mother plant has prepared, and is taking these nutrients in there and allowing it to go through that tissue to reach its source. And it's controlled in terms of directional by a number of factors, and I don't usually talk about it because it's probably more complicated than we actually know. And it's done. So it's sitting on there, it's going to feed it and grow it. Filling and senescence, the last stage. Grain filling refers to the process of cell proliferation and cell expansion, as well as the process of photoassimilate interconversion and starch accumulation. All that means is that it takes the photosynthates from the cells that are something and then converts it into starch and sugar. Grain filling is the final stage of growth and the most important stage in the cereal life cycle. It directly determines the grain weight, grain composition, quality, and final yield. Grain filling is the final goal of the plant. It's no use growing this whole plant and not filling the seed. And that's sort of a sink source relationship, which we'll get into in terms of the importance of that. And as I mentioned, you got to get this plant loaded by the time you get there, because the sink is now going to be the seed. So this is going to start losing its ability to photosynthesize. The leaves are starting to get old, and the plant is going to break that down. So here we are. What are the sources? The photosynthetes and the remobilization. The plant doesn't give up total support of the roots, so it'll still be photosynthesizing. In fact, the actual seed head itself can photosynthesize on those lamellas and so on, because the seed head is the sort of the last thing to actually lose its greenness. But really about 70% of the nutrients that go into the seed come from remobilization of all this accumulation of leaf, stems, and roots. The sink are the roots, the seeds, the fruits, and the tubers. So the roots in the beginning were the sink for production, but in the end, they're the source, not the sink. So the sink changes in the life of the plant. And if you just keep the concept of source sink in mind when you're looking at a plant through its growth cycle, it'll tell you where the important parts are and what's going on in the plant. And our objective as farmers and agronomists is to support that plant and plant health and protect it and give it the nutrients in places it can, we can, and then the plant knows what to do. So here is the chloroplast, which holds about 70% of the nutrients in the cell. It's right in the chloroplast. These are the light sensing materials, the ATP synthase, which is important for ATP production. We talked about it. Rubisco is 20% of the nitrogen in the chloroplast, and that's the enzyme that is used to convert the CO2 into sugar. It's the base enzyme. And there are 16% in other things, and then there's 7% in protein synthesis and cell walls and so on. So the plant has got a lot of nutrients in them, but 75% of it resides in the chloroplast. So it's got to kill the chloroplast, for sure. And during the season, when you get deficiencies, the plant will kill the old chloroplast on the lower leaves and move the nutrients out, along with phosphorus and magnesium, as we talked before. So how does it do that? The process of delivery of cytoplasmic components such as protein and organelles to the vacuole for degradation is now widely recognized as autophagy. Remember this word. You haven't heard it before. This is going to be the big thing going forward. Autophagy is an evolutionary conserved system for bulk degradation of intercellular components in eukaryotes. That includes us. If you're into health and so on, you will find that autophagy is a big thing now in terms of the body scavenging weak cells, old mitochondria, and getting rid of those and recycling them. So here's what's going on. You can read the rest of that if you like. Here's what's going on in the cell. We have the mitochondria, we got the chloroplast, we got the peroxisomes, we talked about all that, and the vacuole. So here's what happens during photosynthesis. It's working all of these things and putting stuff in. But as it gets older and we get into senescing, you get the mitochondria weaken. The chloroplasts are affected by reactive oxygen species because now the chromoplast and the coloring is going and the chloroplasts are being damaged. Those recycle and the proteases go into the vacuole. The vacuole can then absorb through chlorophagy the actual chloroplast. It'll engulf it. It'll break down the mitochondria and take it into the vacuole, and then the vacuole is able to be lysed and move all of those nutrients out of the plant. And that's what autophagy is all about. I sometimes call it autophagy. It seems to be easier than when you're thinking of chromophagy compared to chromophagy. Anyhow, that's another issue. So after it does that, we have this going on in the leaves, for example. We can have, here's the sink and there's the source. So the source is the leaf and it's giving nutrients to the root and the seeds. Okay. Uh, and that's what happens after it wipes out uh, the nutrients, the chloroplast, the mitochondria on the whole in the leaf and extracts all the nutrients out. So it does this all season long. Sometimes it's sending things in, sometimes the root is sending things out, especially when it's senescing. And we can take a look at this. This is the percentage indicate the percentages indicate the relative decrease in mineral content in senescing seeds during grain filling. So all these percentages, that means there's only, they take out 93% is moved, 89% is moved, 43% is moved. With zinc, 92% is moved. In barley, 70%. So the plant moves everything. People say, well, the plant can't move those transitional metals from the cells. Oh, yes, they do. When it's senescing, they move as much as they can to move it into the seed. Otherwise, what would be the purpose of putting it there other than to support growth? So lastly, here's how that seed develops. Water ripe. This is about 8-10 days after pollination. This is about 20 days after pollination. There's nothing much going on in terms of division of cell structure. It's just accumulating DNA and proteins and so on. Then it starts to develop structure and it goes on to maturity. Let's take a look-see. Here it is. You can see that this still the anther is stuck there. The seed is growing. This is the bush at the end of it, which is sort of where the stigma, the stigma comes off. We can see them growing here. After a while, when you cut into it, you can see now it's forming structure. It's starting to form some structure. You can see that here, and then even in the green stage, the embryo is not mature, but is starting to develop, and all the endosperm is full of proteins and starches. The seed finally matures, dries down. The lemma and palea start to loosen off so we can combine that. And the seed is largely now maturing. So what does it take to increase yield? You got to make more spikes. With wheat, you got to have plants there. It's not like canola where it'll spread. So you need one primary spike, maybe one tiller, two tillers at the most in our part of the world. You need to have those spikes survive. You need to have more seeds. That's the only way you can do this. You got to have more seeds and you got to have bigger seeds. That's all we can do to grow a wheat crop. So you got to do that right from the beginning, and the combination of those are what you get for an end result. Lastly, here's what Taurus provides. We look at when we have an opportunity to provide some sort of a treatment that would provide nutrients to all the plants. Because as we saw in the beginning, when we looked at that canola field, there's variability across that field. When you're putting on a foliar spray, you're giving every plant something, regardless of what's in the soil, whether there's enough or not enough. So whenever you go over the field, I think it's wise to consider putting in some sort of a nutrient package, whether it be at herbicide timing or at fungicide timing to support filling, to support growth in that rapid stage. And at prime, you put it on the seed and you support every seed. You make every seed better, because we know that there are deficiencies in the seed depending on the location of the seed on the actual stem. We also know that when a seed grower, you're using your own seed, when you harvest that crop, you're going across all that field. You're taking seed from places that are well fertilized and accessible to the plant and not. The one thing you can do with a seed primer and putting on these nutrients grow through the season is to provide every plant and every seed with an opportunity. And that's how we figure out what we put into the product. Take a look at prime, NPK, lots of phosphorus, boron, iron, manganese, all the things the seed needs to be efficient in terms of dealing with preparedness for photosynthesis with these transitional metals and to deal with free radicals under stress. With herbicide timing, massive amount of phosphorus. We're growing a plant. The plant needs a lot of phosphorus early because that's when the draw is really strong, and roots, unless they're moving through the ground, have problems accessing phosphorus, especially on their cold, wet conditions. And the micros to support all those activities and enzymes. When it comes to filling, it's moving on later on. We don't need all of those transitional metals like we did before. We need some nitrogen and we need lots of potassium. Potassium does what? It controls water. It moves things. It pressurizes cells. It'll move those nutrients from the senescing seeds, or the seeds that are being autophagized or destroyed. We need to move that to the seed, and that goes up through the stem, through the veins, out into the stem and up the stem. So the potassium is really key in that, and it's hard to get late in the year. And that's the stories, ladies and gentlemen. I hope you enjoyed it. It was a pleasure to try and intrigue you into the life of plants. I love this kind of stuff, and I hope it adds to your perspective on growing a wheat crop and this wonderful crop we grow here in Western Canada. Thanks a lot and for your attention.